So let's take a look. Oh, we only have 11 slides for that. Okay. So we are all multicellular bodies. So we're made up of many cells, right? Cells are like a community or a factory. We've kind of seen that each of the cell organelles have their own functions, they have their own role, just like each person in the community plays a role. Some play more than one role, and sometimes you have a lot of organelles that play similar role. Metabolism refers to the chemical reactions in a cell, okay? Because we're talking about this on a cellular level. It's the chemical reactions in a cell. Enzymes speed up the metabolic reactions, okay? So enzymes are for speeding them up. We have other substances that slow down reactions, but enzymes speed them up. Okay, what else do we have? So now, okay. So now we have different kinds of metabolic reactions. Anabolism, which is to build up, okay? Building cells up requires energy for little cells to join with other cells to make bigger molecules. Little cells make bigger cells. Anabolism. And then dehydration synthesis. We watched a video on it, but it might be worth watching again because the type of, well, okay, so there's dehydration synthesis, but it depends on what you're putting together. So if you're putting together sugar, that's one kind of product you're going to get. If you're joining together fatty acids, you're going to get triglycerides, joining glycerol to a fatty acid. Joining amino acids, because remember the amino acids are the building blocks of proteins, you're going to get proteins. Joining together nucleic acids, which are usually proteins, not always, but joining together nucleic acid gives you nucleotides, and H2O is usually another product of such reactions. Now let's stop here for a second, and I'm going to go back to something. See what we have. Okay. So now catabolism is the opposite of this. Okay. Catabolism is to destroy, and that produces energy. So hydrolysis does the opposite of this. So this is the opposite reaction, which is the second half of the video that I just showed you. And I'm going to get you the video in a second. But because we'll go back to that part. But we're going to watch the second part. This part is Where did I put it? Oh, there. Okay. So the second part of this, by the way, this is, okay, so let me do this like this. And I'm going to call that glycogen. Glycogen, because that's what your instructor said. Okay, so now this type of reaction here, the dehydration synthesis, this is, I'm gonna make this bold. Where am I? It's not where I want to be. Where is it? There it is. So this is anabolism. Okay. This is anabolism. And that's the building up of molecules, which requires energy. Okay. Now, the next part of this is catabolism, which is breaking down molecules and that's by a process that we call hydrolysis. And that's a type of chemical reaction in which larger molecules are split into smaller molecules when water is brought in to the reaction. So again, we have 4 parts of this, okay? So this is just the opposite of what we saw before. So here with carbohydrates, glycogen breaks down I thought I was sharing the screen, but I wasn't. So let me, it's okay. No problem. I thought I was sharing the Google Doc. So with carbohydrates, glycogen breaks down into saccharides, into simpler saccharides. So with proteins, polypeptides break down into amino acid. With lipids, triglycerides break down into fatty acids and glycerol, and with nucleic acids, nucleotides break down to nucleic acid. So it's just the opposite of what, and you could call this polypeptides, or you could call this because the other proteins break down into amino acids. So it's the opposite of this reaction at the top. Now the video, I'm just going to give you the link because we already watched the video, and all this does is reverse the process, but I wanted you to have that. So this is the video comparison of of anabolism, actually dehydration synthesis and hydrolysis. Okay, so there is that for you there. Okay, so now let's go back. I'm going to close that. I'm going to stop sharing that. Go ahead and put your slides up again. Okay, so that was just what we went over. Now, enzyme actions are what we're going to talk about next. Remember, enzymes speed up actions, okay? So Okay. Okay, so enzymes speed up reactions. It lowers the activation energy. It lowers the energy that you need to get started on a reaction of some kind. Okay. Because otherwise, most reactions would never occur because you wouldn't have enough energy to start them. Okay. So it changes the substrate. Substrates are the things that we put in. It changes the substrate into a product. Enzymes are not consumed in the reaction, so they can be reused. They can be used more than once for more than one reaction because they aren't consumed in that. The active side of the enzyme twists to fit the substrate. It lets go to fit again. Each cell has hundreds of different enzymes. Okay. And the different enzymes go along different regulatory metabolic pathways. Okay. There's also coenzymes that work together with each other. And enzymes, as we know, can be altered by extremes. Do you remember what alters enzymes? There's about four things that we've talked about that can alter the way reactions work. Do you remember? It's the same kinds of things that will denature proteins.
I think one of them is temperature.
The temperature, right. Temperature will affect enzymes. What else?
I think pH.
Right. How acidic something is will do it. What else?
I think energy.
Radiation will like certain kinds of enzymes, yeah, energy or radiation, sunlight, things like that can alter the way enzymes work. So temperature, heat, temperature, radiation, pH, and energy levels, right? So those are the big four. There's other ones. And sometimes enzymes can be influenced by other enzymes, which is what coenzymes are for. Let's see what else he says in here. What else does he say in the slides? Okay, so this is talking about, oh boy, the Krebs cycle. Yeah, we had a video on it. And this is going to take some study. This one is going to take some study. So looking at the Krebs cycle, excuse me. So, okay. So this is actually three processes, okay, that you're seeing on this slide. There's glycolysis, okay? There's glycolysis, and then there's the Krebs cycle, And then there's the electron transport chain and oxidative phosphorylation. So if you scroll up just a little bit, you should have a subtitle on this. Like scroll up just a bit. Oh, it doesn't have it. That's interesting. Okay, so I'm going to have to break this apart. We watched one whole video on the whole thing, and it's a lot, okay? So what I'm going to do is I'm going to turn this on. So now we have this is another so again, this depends on what you're making. So this is lipogenesis, which is how we get the beginning of fats, carbohydrates, and this is lipids. Okay, so he's breaking it apart. Here's proteins, and this is the oxidative phosphorylation. So this depends on what you're making, okay? We were focusing, what I just showed you, like I told you, this is a lot. What I showed you was, let me annotate the screen. Give me a second. So this, okay. So this part in the middle is what we just went over. That's assuming you did carbohydrates, okay? If you do lipids or you do some other form of reaction, you'll get a different type of, so ours went from glucose to pyruvate to acetyl-CoA, right? And here, this goes from fatty acids to malonyl CoA to acetyl-CoA. If it's lipids, it goes from fatty acids to acetyl-CoA. If it's proteins, it goes from proteins to amino acids to glutamine, glycine or serine to glutamate to ketoglutarate. But this is all oxidative phosphorylation. So that really depends on, what I showed you was glucose, but there's also other ways to do this, okay? And that's what this shows you. This is all oxidative phosphorylation. Okay, let's go on to the next slide, because that could have been a whole lecture by itself. So cellular respiration, okay, this is a summary of what I just spent the last 20 minutes showing you, or maybe more. Glycolysis is the first phase, where a 6-carbon glucose is broken apart, it's catabolized into 2 3-carbon pyruvic acid or pyruvate molecules, okay? Then in the cytoplasm or the cytosol, there's an anaerobic reaction. It doesn't need oxygen, where each of the pyruvate molecules enter separately into the mitochondria. And your net result as far as energy goes is 2 ATP and a bunch of enzymes, okay? Then you go into the citric acid cycle that takes place in the mitochondria, where the pyruvic acid is converted into acetyl-CoA. It's aerobic, so it does require oxygen. You get 2 more ATPs. After that happens, you get oxidative phosphorylation through the electron transport chain, which is where you and I left off. So let's keep going and see what he says about the electron transport chain. Okay, so he's not going to talk about that. He's going to break down glycolysis some more. Okay, so glycolysis is, like I said, it's a chemical reaction that takes food energy that we consume as humans and makes ATP from it. Remember, ATP, he's reminding you, is a 3-phosphate molecule, adenosine triphosphate. So he's reminding you that it's 3-phosphate molecule. Okay, keep going. The Krebs cycle, this is what the Krebs cycle looks like in a slide. So you have all these carbon molecules floating around, okay? You have NADH and FADH2 that are created. So you have this, here's your CoA up here, and you have your NADH. So it goes from your CoA into oxaloacetate. Took me a minute to get that. Oxaloacetate to NADH. It loses some oxygen, becomes a 5-carbon sugar, then a 4. 2 of the 4 carbons, right? Then with the addition of FADH2, you have the 4, and then it repeats. Okay, but it's basically breaking down the pyruvate molecule so that you can create additional ATPs. Okay, then what? What does he have after that? Is that it? Or there's one more? Okay, and then here's a picture of the electron transport chain, which doesn't really tell you much. It just shows you how this is taking place in the mitochondrial membrane. It's showing you how these hydrogen atoms cross the membrane. These are the enzyme pockets that allow it to cross, and the electrons go across because they can't cross the membrane by themselves. And you get a lot of positively charged hydrogen. Okay. So that's where that's coming from. I'm sure there's more detail coming with that, with each of those phases. I'm pretty sure that he's gonna go into that more, or he'll have a lab on it somehow. But yeah, I gave you some notes on the Google Doc, so that should help somewhat. But just remember that the glycolysis has two phases basically, and then there's that intermediate phase that you get acetyl CoA from. Then you have the Krebs cycle, and then you have the electron transport chain. So those are separate phases of the reactions to make food energy. Okay? And that's the end of that. Let me see if there's a I think we have a little bit of time left. let me see if I have some.